So let's switch to English right now. Welcome everyone who has joined this seminar today. So today we are glad to have here with us Dr. Marco Ataide, who kindly accepted our invitation. So thank you so much, Dr. Marco. So to start with, I will just do a brief introduction of you, so then you can start the presentation, okay? So Dr. Ataide has a bachelor in biological sciences from the Pontifical Catholic University of Minas Gerais, and a master's and a PhD from the Federal University of Minas Gerais, where he worked at Professor Ricardo Gazinelli's lab that he concluded in 2014. So then in 2015, he moved to Germany to do a postdoctoral research at the University of Würzburg at Kerstin Miller's lab, working with the biology of T cells. So in 2020, he started a postdoctoral fellowship in Professor Dario Zamboni's lab until 2022. And then he moved to San Francisco in California, where he's working right now in AMGEN as a senior scientist and a group leader in the inflammation department. So his current research focuses on the study of autoreactive T cells and the myeloid compartment in the context of autoimmunity. And it's about this that he's going to talk a little bit for us today. So Dr. Marco, thank you so much again for being here with us. Feel free to start the presentation. Just remember to the audience that we are open to questions at the end of the presentation, okay? Thank you so much, Professor Marco.
Great, thanks, Luisa. Thanks for the invite, and Professor Gustavo as well. It's a great pleasure to give a talk to people in São Paulo, in Rio. Those are the best, one of the best places to do research in Brazil. And to me, it's a great pleasure. Yeah, as you said, I'm currently working as a senior scientist at AMGEN, which is a big farm. It's located, I'm here in that building in South San Francisco, like 10 minutes from San Francisco. And AMGEN's head square is in a sit down here. So I'm here, and this is the head square in Thousand Oaks. It's a huge company. It's great for fresh PhD who wants to perform or to have their career in industry, doing cutting edge research, in some of the discovery bubbles, some translational research. So I'm currently located at the Department of Inflammation Immunology, doing discovery research and in autoimmunity, as you said. So unfortunately, I can't share any data so far because of the rules, but I hope in the near future I can share some of the data that I already got here from AMGEN, some basic and translational research data set. So, but what I'm going to share with you today, guys, is a couple of data, a couple of results that I got from my postdoctoral training in Germany. So dealing with T-cell immunobiology and how fundamental research in this topic could help us to develop better cell therapy and vaccine design. So yeah, so I've been in Germany for a couple of years, for almost 6 years, from 2015 to end of 2020, when I went to Dario's lab. So I first went to University of Bonn, which is such a beautiful city, and then a more international city than Würzburg. Würzburg is a small town, but there I joined the Max Planck research group. It's very close to Munich. It's also beautiful. Plenty of vineyard, so it's a great place to be as well. So and one of the stories is dealing with the molecular basis for CD8 T cell memory development, and the second one will be very basic and fundamental concepts in unconventional T cell immunobiology. So when I as you said, I've been work with Professor Gazinelli before. Starting my postdoctoral training. So I was trained in innate immunity. And then when I moved to Germany, I had to learn everything from scratch, the adaptive immunity part. And I got very much interested in that beginning of, I mean, before that, this is a classical curve of, let's say, CD8 T cell immune response that I guess most of you know that. So you get the activation, and then the cells expand and they contract later on because it's a huge amount of cells. You have to get rid of them after fighting the virus or any invaders or tumor cells. Later on, you get a memory pool. And then I was very intrigued by the signals that those cells are getting at the beginning of the activation, even before expansion, because it's very well accepted that a lot of things that happen here at the beginning have a big impact later on in the memory compartment. And as I said, as an immunologist, I had the chance to review the literature again and come up with fresh new ideas that people in the field probably just didn't pay attention on it anymore. So then I was looking for some public data set back that single cell sequencing analysis, and I was combining different source of single cell sequencing data, looking for some molecules that would be expressed in different phases of the CD8 T cell activation, but especially at the beginning of the activation. And I found a couple of molecules. And one of them, and then I want you to pay attention to that region here. This is day 3, transition to day 4. I found a molecule called BATF3. And BATF3 is a transcription factor that I'm going to tell you a little bit more in a couple of minutes. But that was really interesting because that molecule are not expressed in naive cells, and then they get expressed in T cells that get activated by day 3 and day 4, and then disappears. And then even in memory phase, that molecule never comes back. So then I tested myself in vitro to see if I could see that molecule. And in fact, in vitro, a few hours after, like 48 hours after activation, we see a peak, and then it's gone, so we don't see it anymore. Also in vivo, when we had antigen-specific T cells getting activated, here was OT1 T cells. As you know, those cells recognize ovalbumin. And then after activation with a virus expressing ovalbumin, a couple of hours after, I could see BATF3 expression, but 72 hours later, I didn't see it anymore. So that's a very transient expression. And then I was very, very intrigued by that. I said, okay, that might be something interesting to look. So as I said, BATF3 is a very important transcription factor as a member of AP1 family, which is, again, another important family of transcription factor. So that's the classical AP1, Jun and Fos. It's a heterodimer. BATF3 can also be paired with Jun and compete for Jun with Fos. And in that way, people in the past thought that's a regulator molecule. This transcription fact is associated with cytokine production, survival, cell proliferation. So a lot of aspects in the cellular process that this molecule are involved. So that's an old paper from 2008, but very important one from Kenneth Murph lab, showing that the deficiency in BATF3, so the full deficient mouse has no DC1, the CD8 alpha positive dendritic cells. And of course, in this full knockout mouse line, the CD8 T cell immune response is compromised because one key component of the immune response is missing, the CD8 T cell, sorry, the CD8 positive DCs. So then I was speculating that people, and also based on this transient expression of BATF3, I started speculating that people might miss the role of BATF3 intrinsically in CD8 T cells, because when you use the full knockout, you simply don't get the proper immune response because there is no DC. So it's easy to overlook that role in CD8 T cells. All right, so you're gonna see during my presentation that I use this setup doing called transfer of wild type and BATF3 CD8 T cells into hosts. So sometimes I'm not gonna show that schematic, so you can assume that is when it's in vivo, it's a cod transfer, but I will remind you. So that's the classical way to study antigen-specific CD8 T cells, right? Transferring naive T cells and then immunize the mice, and then later on you can take a look during the activation phase or the effector phase and the memory phase. So here I transferred wild type and BATF3 knockout CD8 T cells, and both are over antigen specific. So after transferring the cells, we infect the mice with Listeria expressing over. And then at day 8, I checked how the cells look like in terms of expansion. So, and then 30 days later, I also had a boost. This is a heterologous protocol. Now it's vaccine expressing over. And then 6 days later, I was checking the recall, the memory recall. All right, this is the day 8. When I compared the wild type in BATF3 T cells in terms of relative frequency, also functionally, I have checked the cytokine production. I don't see any difference. That was a little bit disappointing, but yeah, that's the way it is. However, when I check at the recall phase, I saw a dramatic difference comparing the wild type and the BATF3 knockout. So it seems that the BATF3 knockout CD8 T cells, they can easily get activated, they expand, but something is going on that during the memory phase, I can't recall those cells properly. So yeah, the question before that, so the question is, because here you could say, yeah, so is the size of the memory pool, or those cells cannot be recalled because they haven't a defect? This is just a number game, or just a quality problem? So then I performed the kinetic study. So this is day 0, 55th, and then over time, what we found was that the BATF3 knockout CD8 T cells during the contraction phase, they crash, and then we have a very, very small memory pool of CD8 T cells compared with the wild type. All of this were in the same mouse. That's important to mention. This is blood. I've checked. I don't know if it's here I look, but I check in lymph nodes, spleens, it's everywhere. So we have a very poor memory pool of CD8 T cells when they don't have BATF3. And remember, the expression of BATF3 was just at the beginning. This is something that some inputs that the cells get at the beginning and has a big impact later on during the memory phase. So then I decided to sort these cells because it's clearly a problem with the number of cells. If you have less cells and then you're now going to recall, as I did, it's very likely that you get less cells compared to the one that you have more cells at the beginning. So then sorting those cells and put it again in a 50-50 ratio to give the same chance to expand, I still see less BATF3 CD8 T cells compared to the wild type. So it's not only a number game, but also a quality issue here that BATF3 deficiency leads to. So yeah, that's it. So we have a small size of memory pool, and the capacity of those cells to be recalled is also smaller. All right, so to make the long story short, because I have another story to show you, we tried very hard to understand what is going on when BATF3 is absent. And I think it's a couple of things. It's the whole genetic or the transcriptome network changed. But the one thing that was really dramatic was the, let's say, inefficient BIM, which is a pro-apoptotic molecule downregulation. We noticed that after activation, CD8 T cells, they downregulate BIM, as you see here in gray. But the BATF3 knockout CD8 T cells, they cannot do it in the same extent. So that would be one of the explanations for the very strong contraction that we saw at the beginning of the contraction phase, and then the end we have a small memory pool. So then we decided to knock down BIM in BATF3 deficient CD8 T cells. So we use the shRNA system to knock it down. And here I'm comparing BATF3 knockout CD8 T cells that were treated with the scramble. Nothing was happening, just a control shRNA and the wild type scramble. As expected, this is like wild type versus BATF3 knockout. We get more wild type cells compared to the KO. On the other hand, when we knock down BIM in the BATF3 deficient CD8 T cells, we could reverse the phenotype. So now, for the first time, the BATF3 deficient CD8 T cells are out competing their counterparts, the wild type CD8 T cells. All right, so that was the loss of function, and we have done RNA sequencing analysis on those cells, and in the apoptotic pathways was the most prominent pathway in that sequencing analysis. We believe there are many things involved, but the cell death and apoptosis was the most important aspect. And in fact, as I showed you, we could rescue the BATF3 knockout CD8 T cells simply knocking down this pro-apoptotic factor named BIM. So then we decide to switch gears and move to the gain of function approach. We decide to overexpress BATF3 in CD8 T cells. And now what we see is a higher downregulation in BIM downregulation expression, and compared to the wild type CD8 T cells. They are doing this, but now with overexpression of BATF3, they do even higher. So, and doing the same experiment setup that I showed you initially, transferring wild type, and in that case now BATF3 overexpressing cells into mice, and then challenge them with a vaccine over. For the first time, we now see the wild type cells not doing so good compared to the other cells, which now are BATF3 expressing cells. So we clearly could give cells an advantage when BATF3 is sustained. If you remember, BATF3 was transiently expressed only at the beginning, but now, modifying the T cells, we can give to those cells an advantage and they survive more. It's important to note that when BATF3 is overexpressed, we don't see more cytokine production or granzyme expression. The factor capacity looks very similar, but what seems to happen is not only the capacity to survive is there, but the stamina capacity of those cells is higher. I have tested, I'm not showing today, but I have tested, for example, molecules associated with exhaustion like TIM3, and they express much less, and they express higher levels of TCF1, which is a good marker when the cells are exhausted. So those TCF1 positive cells, they can be rescued from the exhaustion situation. So it seems that change again the whole network controlling the immune response. But one important thing that came out was the survival capacity, reverting completely the problem that BATF3 deficiency was giving to those cells. So here is another approach, as I was mentioning. we saw some exhaustion markers getting downregulated. So then we decided to put those cells, the BATF3 now overexpressing cells, in a situation of chronic antigen exposure. So mimicking, I use the virus infection, I'm going to explain the model to you, but this is the situation that CD8 T cells see in a tumor environment. So here I use the LSMV, the chronic strain, and I infect the mice for 30 days, so to get a chronic environment. And then at day 30, we transferred wild type. In that case, here the P14, they are not OT1 anymore because the P14 is the one recognize the antigen from that SMV. And then the BATF3 of expressing P14 cells. This is the input, day 0. I mean, day 30 after infect, but now the cells they were fresh. Then 7 days into that environment, so 37 days post-infection, I recovered that amount of cells from spleen. And now when I compared the relative frequency of BATF3 and wild type, I can see that the overexpress of BATF3 gave a big advantage to those cells. And they produce normal amounts of cytokines, and they are just happier compared to the wild type. So we see that BATF3 has a potential for adaptive immunotherapy. So, and then just to go a little bit further, we decided to do this in human cells. And again, that BATF3 that we are overexpressing here, the mouse BATF3, and then we put them in human CD8 T cells. And this is in vitro, of course. This is some CD8 T cells we got from PBMCs. And this is the input, and this is over time. You see that the wild type cells, they disappeared compared to the BATF3 overexpressing cells. The BATF3 overexpressing cells are just expanding much more that you don't see much of the wild type. So then we believe that molecule could be further explored in the context, for example, of CAR T cells. In fact, some of our collaborators of this work are exploring that molecule in that regard, in the context of a CAR T cell. So, but I just came across that I found a couple of papers recently that is related to that story that I'm telling you. So this was a paper published in 2018 showing a study case of a patient that got CAR T cells to treat leukemia. And this patient was in complete remission. And then when they look at the CD8 T cell compartment, they found that at the peak of the response, almost 100%, I think it was 95% of 94% of those cells, CD8 T cells, came from one single clone. That was super weird, but very, very interesting because they thought, and in the end, they still believe that this complete remission was due to that phenomenon. And what they found, so after sequencing this cell population, they found that TACH2, which is an epigenetic regulator, got disrupted when the virus introduced the chimeric antigen receptor. So that was really cool, and people since then started speculating that manipulating TACH2 would be a nice way to get better CAR T cells in situations that the CAR T cells are not surviving much, because in that work they saw that those cells, they proliferate, they hyper-proliferated. So they believe that was one of the reasons that this patient got a better outcome compared to the other ones. And now in 2023, what people found was that TACH2 actually restrains the BATF3 expression. So what happened is when TACH2 is disrupted, So BATF3 goes up, this is BATF3, and this gives to the cells a big advantage in terms of survival. A function, again, it doesn't go super up. Actually, in that study, they've shown that the functional capacity, the amount of cytokine and all the effector molecules, a little bit downregulated, which is not bad because then we avoid toxicity, right? So that was very, very cool to see that some of our initial finds based on our fundamental studies are actually now being used to help patients. All right, so this paper, this work has been published in Nature Immunology 2020. So a lot of details that I didn't mention today are there. If you have some additional questions, you can also go there. There is also a very interesting news and reviews summarizing the story, which is very simple, as you saw, but with a big potential. So here's the take-home message. So BATF3 is transiently expressed in CD8 T cells at the beginning of the activation. And then if the BATF3 is not there, so if the cells are deficient for BATF3, you have a normal expansion of CD8 T cells upon viral infection, for example. But during the contraction phase, those cells, they contract much more than expected. It seems that BIM is not efficiently downregulated, and then the cells, they just die off. And then you have a poor memory pool. If you need to get those cells recalled, you're gonna get a problem. So on the other hand, when BATF3 is induced, we increase the memory capacity and also metabolic fitness that I didn't show you today. And also BIM is downregulated, and you get a bigger memory pool. Also in the context of LCMV or chronic antigen exposure, those cells get a better advantage compared to their counterpart. All right, so now I will still have some time. I'll tell you another story. So that was the part of conventional T cells. And now I switch gears because parallelly, I had the chance to explore the unconventional T cell immune response in the context of, in the steady state and in the context of bacterial infection. All right, so that's the classical cellular classification, right? So we have the myeloid leukocytes, lymphoid leukocytes, and it's easy to us to understand when those cells are also classified as part of the innate immune system and others of the adaptive immune system. So we have here, you know, your favorite cell type, macrophage or disease. On that side, our CD8 or CD4 or B cells. And then in that interface, we have a group of cells that we call, together with NK cells, that we call unconventional T cells, gamma delta, NK T cells, mate cells. And there are also other cells that we don't know actually how to classify them, because for all of this, the way we use to classify those cells are mainly, I mean, ontogeny helps, but also TCR sequence. If you don't know the TCR sequence for this kind of cell, it's hard to define them, or the MHC restriction, that kind of MHC restriction those cells are, if you don't know how to classify. So there are some unconventional T cells I can tell you that we don't know the name of them. So unconventional T cells, they develop in the thymus. They start as naive, and but in the thymus, they also get matured. They are preset, and they leave the thymus, go into the peripheral tissue. When you see those cells in the peripheral tissue, they are very much, or they are similar to a memory, let's say CD4, CD8 T cell. They have a memory phenotype. They are activated, and this is very different compared to the classical conventional CD4 that they leave the thymus as naive state, and then they go to the tissue, let's say the lymphoid tissue, they get matured, and then they become Th1, Th2, Th17, or all the Th we have out there. So that's a big difference comparing the conventional and unconventional T cells. And then the unconventional T cells, they also appeared in the lymphoid tissue, and people don't know much what is the relationship between the peripheral tissue and the lymphoid tissue. In that work, I'm going to show you, we saw or we defined that those cells are constantly migrating from peripheral tissue and shaping the local immune response in the lymphoid tissue. Some of those cells, they get activated. I'm not going to show you here. They leave the peripheral tissue, and sorry, they get expanded. They proliferate highly rates, and then when they go back to the peripheral tissue, and that's something that we don't understand quite well, but this is happening for sure. All right, so if you get lymph nodes, different lymph nodes, skin drain lymph nodes here in red, or mediastinal lymph node, or mesenteric lymph node, and in steady state, and you stimulate those cells in vitro here with this very common stimulation system, very unspecific PMA ionomycin. You see that in some lymph node, for example, you have here in the skin drain and the mediastinal lymph node, we have a good amount of IL-17, but not that much in the mesenteric lymph node. Remember, this is steady states lymph nodes. GM-CSF seems to be highly abundant in the mediastinal lymph node, but a bit less in the other ones. Interferon gamma in the skin drain lymph node is more there than in mediastinal and mesenteric. You have difference in terms of cytokine and signature cytokine comparing different lymph nodes. And more than 75% of the cytokines in steady state, when you have this setup, are made by unconventional T cells. And just to simplify here, unconventional T cells, I'm calling CD3 positive, CD4, and CD8 intermediate double negative. We know that some NKT cells, they can also express CD4, a bit of CD4, especially in humans. MAIT cells in humans, they can also express CD4 and CD8. In humans, everything is more complicated. But in mouse system, we can safely say that we have the CD3 positive, CD4, CD8 intermediate to double negative. I'll show you some gating strategies. All right, so we decide, so based on this, that's interesting. So we have a regional immune response. Let's try to explore a little bit more this. Then we decide to infect some mice subcutaneously with Staph aureus, and then look at the unconventional T cell immune response there. What we found was, this is day 3 post Staph aureus infection, was a great expansion of gamma delta T cells. Actually, here in that gating strategy here, we didn't know which population was, but we noticed that in the TCR gamma delta knockout mice, that population is gone. So it's very likely that those cells are gamma delta T cells, and in fact, so this is a clonal expansion. Mainly, let's say 9% of those cells are gamma 6 positive. So then we say, okay, let's take a look in the mediastinal lymph node, and in fact those mice with Staph aureus intranasally. And this is what we see. Comparing to the skin drain lymph node, we don't see such expansion. I'm not showing you here, but we don't see any of those Ki67 positive cells. And you see that this clonal expansion based on the V-gamma 6 expression, it's also not there. But let's say the V-gamma 6 positive cells, not the expanded ones, exist in the mediastinal lymph node. They simply don't expand there. And it's important to mention, I'm not showing here that we of course, we made sure that the cells in the mediastinal lymph node saw that bacteria. So, for example, if you check the conventional T cells activation, we see a bigger difference compared to the unconventional. Was some things particular with the unconventional T cells that some of the cells could expand in the skin drain lymph node, but not in the mediastinal lymph node. All right, so here's another example. So this is again just to show you the difference between lymph nodes and the immune response depending in which lymph node you're talking about. Because in theory we think that the lymph nodes are the same and the cells should respond in the same way, but it's not true, at least for unconventional T cells, but it's true for apparently conventional T cells. So now we used another bug. We used the vaccine virus. And we did the same, sub-Q infection and intranasal infection. Now we are measuring L4. Remember that we saw in this PMA Iona experiment a great amount of L4 in the mediastinal lymph node treated, but not in the other lymph nodes, or much less. So in vivo, it seems to be exactly like that. So here's in red is the amount of the frequency of L4 positive cells in the skin drain lymph node, and in green is in the mediastinal lymph node. And this is a frequency, and this is absolute number. So it's again showing that we have a regional heterogeneity in terms of immune response based on UTC biology. So, and now we see that the cells that releasing that cytokine are mainly NKT cells based on the CD1D tetramer signal that we see here, and here's some of the gamma delta. Here are some of the mate cells and other cells that you don't know how to classify. So the main source of L4 in that setup is NKT cells. So in fact, a very nice paper from Facundo Batista's group, published a couple of years ago in Cell, have shown that NKT cells are important source of L4, and those cells just initiate the germinal center formation. We know that the TFH cells, they help a lot in terms of, also in terms of L4 production, but in this work, they claim that without the NKT cells, the germinal center is much smaller, we have less cells. And then we decide just to look, because now we see NKT cells behaving differently in the mediastinal lymph node compared to the skin drain lymph node. So here we go. When we use the CD1D knockout mice, full knockout mice compared to the wild type, and then checked for the germinal center B cells here, classified as fast positive and IgG1 positive cells, we see a reduction in the CD1D knockout mice. So this was in the mediastinal lymph node, in agreement with Gaia's paper. So then when we checked at the skin drain lymph node, we see now more of those germinal center B cells in the CD1D knockout. And this is weird because here we had less L4 compared to the green. So anyway, but that might be something that we still don't know, other cytokines that could help, but we didn't really follow this. We just want to point it out that we have difference in the lymph nodes in terms of adaptive immune response, directly as we've seen for the clonal expansion of gamma delta T cells, or indirectly, as we are seeing now in the absence of NKT cells, we have completely two different outcomes in two different lymph nodes. And then we're going to discuss more about it later on, but that call our attention to the fact that sometimes it's important to think about how we should deliver vaccine antigens. So what is the main site for that particular infection, as we have now, for example, for COVID, right? Maybe intranasal. If some people tried vaccine might be more interesting than intramuscular. Anyway, that's something that was there, and we saw this heterogeneity in terms of immune response comparing different lymph nodes. So to better understand this, we decided to do a single cell sequencing analysis. Then we put 3 different lymph nodes, 3 different kind of lymph nodes: mediastinal, mesenteric, and inguinal lymph node. We add some hashtags on those that later on, after sequencing, could discriminate from which lymph node the cells we evaluating are from. So we mix the cells together, and then we sorted. So this is the CD3. We gate on CD3 positive cells, and as I said, CD4 and CD8 they are here, and we call intermediate negative cells. Those are the cells sorted and hashtagged and sequenced. So we found 2 bigger clusters, and every cluster here. So here are the 2. We have small clusters. So one of the molecule, one of the gene that was differentially expressed comparing those clusters was the CD6C12. And as you know, this molecule is very important for the cells to recirculate through the blood and getting to the lymphoid tissues. So then we start saying, wow, that's interesting, because it seems that we have a compartment, so the cell negative, that might be lymph-borne. So the cells that might coming from the peripheral tissue and go into the lymph nodes. And we have another group of cells, the blood-borne, that they are constantly recirculating. And then we thought that this population here is the population associated with this local immune response and the difference between the lymph nodes. So interestingly, you see that now in the blood-borne, you see that all the cells, as I said, now we have the chance, based on the hashtag, to discriminate from which lymph node these cells we're evaluating are from. So here we have the inguinal, mainly a mediastinal and a mesenteric lymph node up here, which is a little bit mixed with the mediastinal. But in the blood-borne, the one that we call in blood-borne, they are all mixed together, which makes sense because they are recirculating through all the different lymph nodes. And these cells here, they are probably coming from the peripheral tissue. So then we decide to focus on that particular idea and in this particular phenomenon to try to prove it and see what is the consequence of having cells coming from the peripheral tissues. Yeah, so here's just basically the same thing here now, but now they are separated. You see that the inguinal lymph node are mainly, we have cluster 3, cluster 6 mainly, mediastinal cluster 2 and cluster 4, and mesenteric basically cluster 4. Of course, there are some sharing here, but you can clearly see that the lymph nodes are different. And what is funny is we see all of the all of the unconventional T cells, gamma delta NKT, mate cells, which are TCR, they have difference in the TCR, but they don't cluster apart. So classifying cells based on TCR, I don't know, it might not, I think it's valid, but maybe, doesn't tell you that the cells are really different. So here we have all of the different kind of unconventional T cells, and they are in the same cluster. All right, so the same picture, you see the blood-borne, all of the different clusters we have here from 0 to 8, they have a part of the number 8 that have less mediastinal, but in all of them you see more or less the same amount of cells coming from the different lymph nodes, which is quite different from the lymph-borne, of course, because some cells are coming from the tissue, for example, skin cells coming from the skin, you see then more in the skin drained lymph node, but not in the mesenteric lymph node. That's why we have that difference here. All right, another piece of data that lead us to explore those cells as a functional unity is the behavior of those cells. Here I'm gonna just show you a group behavior pattern, which is a classical kind of group behavior of myeloid cells. So in red you have your neutrophils. That site here was hit with a laser, damaged, and then the macrophage comes later after the neutrophils. Everybody goes to one spot. That's what we call group behavior. It's based sometimes in some chemokines or the components that guide those cells. And here in green, we have the CXCR6 reporter mice that was developed to study NKT cells in the liver. But we know that the CXCR6 positive cells here are not only NKT cells. And what we are seeing here is not liver. This is lymph node. This is intravital microscopy. And this was a steady state. Probably something happened here, a cell death. And in red, we have, I'll play it again. In red, we have some conventional T cells, CD8 T cells. And in green, we have the CXCR6, which are mainly the unconventional T cells. And this kind of group behavior, it was never seen in T cells. And again, this is unconventional T cells. So the cells they apparently work as a functional unit as this group behavior suggested. So they're all different kind of unconventional T cells. So as I said, gamma delta, NKT, and MATE cells, why they are behaving as a unity. So then we decided to run another RNA sequencing analysis. So based on the CXCR6 expression, we sorted that population. There was CD3. We also included some of the CD3 negative because we want to have some contamination in our single cell sequencing analysis to help us to differentiate the unconventional T cells. So, and here is the data. So basically, this is the group of unconventional T cells. We have 3 different clusters. And again, what is interesting here, that we find all of the different unconventional T cells in all of the 3 different clusters. So the TCR sequencing here seems not to be important to define that population as a functional unity. Of course, the adaptive immunity might rely on the TCR sequencing. As I showed you initially, in some situations, depending on the lymph node, we have the expansion of one kind of unconventional T cells, but in another situation, you don't have it. So, but now we decide to explore those cells in a different way as a functional unity. That's why we now are focusing on this group of cells based on the CXCR6 expression. That RNA sequencing data also helped us to define another marker. I introduced the integrin beta 7. So we have the integrin beta 7 CXCR6 double positive as a very homogeneous population. But inside of that population, what we get is gamma delta, MATE cells based on MR1 tetramer positiveness. Also, among the double negative for gamma delta and MATE, we have a couple of CD1D tetramer positive cells, which are the NKT cells. And down here, we have other unconventional T cells that I don't have an antibody, anti-TCR, or any other tetramer to stain. So I don't know how to classify them. I'm calling them others. So importantly, remember that our initial RNA sequencing data, we saw CD612 positive and negative. So the population here I'm now calling in gray, the double positive for integrin beta 7 CXCR6, they are CD612 negative. So they might be the ones coming from the tissue. And this is only skin drained lymph node. I'm going to show you how it looks like in other lymph nodes, which is a bit different. So, and the one down here are CD612 positive, or the majority of them. We have some of negative that also probably coming from the tissue, the peripheral tissue from the skin. So we decided then to focus in that population, again, because we are interested in those cells coming from the tissue, defining the regional immune response. So as I said, we had that speculation that the cells are coming from the tissue, and it's very hard to show it. So we initially took advantage of this mouse line called Dendrite II. Dendrite II is a protein, it's a fluorescence protein. Then when you hit this, the protein or the cells that now carry the protein with UV light, those green, they also they become red. So then what we did, we used the Dendrite II mice, and then we hit the skin with UV light, and then wait for 24 hours. So 24 hours later, I was collecting the popliteal lymph node, so the closest to that site of illumination, and then I checked I was looking for red cells, because if the cells are really migrating, I should be able to see some green, also positive for red fluorescence. So here we go. I gated on those cells, integrin beta 7 CXCR6 positive. This is non-illuminated. As I told you, all of the cells are green. So, and here we have some of the illuminated cells. And we see that now in the lymph node, this is lymph node, okay? So in the lymph node, we got a population of red cells. So it tells us that cells are constantly migrating from the peripheral tissue, at least in that setup, in the skin, to the lymph node. And then I use the FTY720. I think some of you might know, if not everybody. This is S1P antagonist, and S1P is it had been related to migration of leukocytes from tissue, also from lymphoid, from peripheral tissue, lymphoid tissue. So that's the way that you can trap the cells in that case in the skin. So that was an additional control for us, that the cells are in fact coming from the skin. So, but what was very interesting is that population that appears here in the popliteal lymph node, they do not exist in the mesenteric lymph node, but they do exist in the skin drain lymph node. Sorry, in the mediastinal lymph node that I'm not showing here. So then I said, well, that's interesting, because that might give us another opportunity to test if those cells are really coming from the tissue, peripheral tissue, and going to the lymph nodes. We decided to make a transplantation. So I took some of mesenteric lymph node, and then I put them, so the cells are not there, right? And then I put them in the popliteal fossa, so where the lymph node, popliteal lymph nodes are seated. So I put the lymph node into the fat of the popliteal fossa and waited for 2 months. So here we go. Again, this is the endogenous skin drain lymph node. This is from the other leg. Here's the endogenous mesenteric lymph node from the mice that got mesenteric lymph node into the popliteal fossa. Again, we don't see the population there. And here is the mesenteric lymph node that was transferred into skin drain lymph node. So the cells are in fact coming from the skin. So then I did a vice versa experiment. So I took skin drain lymph node, and then I put in the mesenteries. Remember, skin drain lymph node, we have that population. In the mesenteric lymph node, we don't. So then when I transfer skin drain lymph node to the mesenteries, that population disappears. So those cells are constantly migrating from the tissue, and at a certain point, they got replaced. So the cells are gone. So if you just check another phenotypic analysis of those cells, he based on CCR6 and CXCR3, just define the Th17 and Th1 signature of those cells. So then the skin drain lymph node, we have more cells prone to be Th17 compared to the Th1. And this is vice versa to the mesenteric lymph node. So we have more CXCR3 positive compared to the CCR6. But now, when as I said, when I put the skin drain lymph node into the mesenteries, so that phenotypic analysis switched completely. So we see that the cells are more CXCR3 and compared to the endogenous skin drain lymph node. That was another evidence that the cells are converted. Sorry, the lymph node are completely converted into mesenteric kind of lymph node. So you see, this is the graph. It's very different from that one. That one is more close to that bar here. All right, so I'm just trying to hurry up here to get it done. I just have a couple of slides to show. So what we found was that population, this integrin beta 7 CXCR6, they are compensated. This is a steady state. So we have TCR gamma delta knockout, CD1D knockout, no NKT cells. This is the MATE MO1 knockout. This requires a specific background literate control because this is another background. That's why it's not the black 6. But you see that in the absence of those cells, that whole population is the same. The size is basically the same. And you have just to compare these two guys, 10%, 13%. So what is missing here in the TCR gamma delta, basically gamma delta, but the population is there. And what is missing here in the CD1D knockout is just the NKT cells, but it doesn't matter, the population is there. And the same thing happens here for the MATE cells. Here's the MATE population is disappearing with the deficiency compared to the CAS, not to the CAS mice. But in the end, we have the integrin beta 7 CXCR6 full population. So, and here's just another way to show. Remember, we had the CD6012 positive and negative, and in terms of relative frequency doesn't change, also number doesn't change. So deficiency in this mouse line, in all of those mouse lines, doesn't really disturb that functional unit that we've been exploring. So that seems to happen in humans. So just quickly here, this is a PBMC from human donors. So this is the MR1 wild type, and this is a patient with a mutation in the MR1 gene and with the allele that's absent, and the patient doesn't have MATE cells. So in the end of the day, what happens here is that population that can produce TNF or interferon gamma are basically gamma delta T cells when they are restimulated in vitro. So the cells, they can just take over and fulfill the niche. So we found that those cells, the integrin beta 7 CXCR6, they are seated here. So here's IL-18 receptor D tomato and IL-23 receptor GFP. We developed this mouse line to show that the cells in the lymph node, they're sitting in the sinus, just in the strategic location to receive some of the TLR or inflammasome-derived signals. As I said, those cells, they are preset, they send cytokines, and they can fight bugs. They can also, in combination with IL-1 beta and 23, they can make IL-17. And some of them, in combination with IL-18 and 12, they can make interferon gamma. So, just to show you a little bit of their microanatomical location and how important those cells can be in the context of bacterial infection. And as I said, if you have a deficiency in one subset, there is no problem because the whole niche is there. But now the way we found to remove the whole compartment, at least functionally speaking, was deleting L23 receptor. So in the L23 receptor knockout. So this is the amount of L17A and L17F among the unconventional T cells. In the 23 receptor knockout, you don't see that. So again, here TCR gamma delta knockout or CD1D knockout or MO1 knockout, you don't see any problem in terms of L17 production. But in IL-23 receptor, we do see. And the cells after sequencing, we also see that they are very homogeneous. So all of the unconventional T cells are very similar in terms of transcriptional profile. So again, pointed out for the idea that those cells are operating in functional units. So, and here, testing the idea of those cells, of the regional immune response, as we know, we have in the skin drain lymph node those cells, the integrin beta 7 CXCR6, and they are prone to produce L17. So when we have a subacute infection, we have a great amount of L17 production. Intraperitoneal infection with Staphylococcus aureus, we don't see in the mesenteric lymph node because those cells, they are not there. So, and again, when we now do the same kind of experiment, we see a great infiltration of neutrophils in the wild type mice, skin drain lymph node, but not in the L17AF knockout. But nothing really happens in the mesenteric lymph node when we have intraperitoneal injection. That's another experiment just to stress more the idea that every lymph node is different. That's the main message of that study. And this is the final piece of data I'm going to show you. This is the bacterial load in the mesenteric in the drain lymph node, which is very tough to get because most of the fight are in the peripheral tissue in those cells. As I said, I'm not showing you the data. They go to the lymph node, they proliferate there, and then they go back to the skin or to the lung or to the gut. So, and again, you don't see any problem with the knockout mice for different unconventional T cell compartments. But when we have a deficiency in the functional molecules, L23 or 17, and then we do see an increase in the bacterial load. But in the mesenteric lymph node, it doesn't matter because nothing is really happening there. So at least in terms of unconventional T cell. All right, so again, this work was published last year in Immunity. And as I said, the message is very simple. We have the unconventional T cells in different peripheral tissues, and they are not the same. They are not the same, and they constantly migrate to lymphoid tissue, to lymph nodes, and there they provide a distinct immune response. So, and this might be important, sorry, some of those immune response can be adaptive immune response or the cytokine dependent as the one that I explored more in the end, or also the TCR dependent, as we have seen initially for gamma delta T cell that happened in the skin drain lymph node, but not in other lymph nodes. So this kind of knowledge might help us in the future to better design or to think more about vaccine design or about immunization route. Cell therapy, especially for cancer, some of those cells have been associated to better fight of cancer, or in some situation they are worsening the anti-tumor response. Also in autoimmunity, depending on the tissue, those cells can be detrimental or protective. So that's again another piece of fundamental work that can have or that has a potential to be translated. All right, so here the most important slide, I would say. So I'd like to thank Professor Castamiller, one of the most creative scientists I have ever met, and the two PhD students that I had the chance to guide, and that helped me a lot for both studies, especially for the unconventional T cell studies, and all of you guys for your patience, and I'm happy to take any additional question. Thank you.
Thank you so much, Marco. Amazing results. Very nice presentation. So now we are open to questions. You already have a question in the chat, so I'll read it to you. Okay. Vinicius Borges is asking, nice talk, Marco. You have such exciting results. I have a couple of questions. The first one is about BATF3 intrinsic expression on immune cell components. Is it better overexpress BATF3 on disease or CD8 T cells to improve cell therapy? And the second one is about tertiary lymphoid organs associated with the immune-excluded tumor microenvironment. So have you ever heard about a BATF3 DC1 independent mechanism of cross-presenting and activating CD8 T cells?
Okay, the first question was asking if it's a good idea to express BATF3 in CD8 T cells to fight cancer. Is it correct?
Yes, that's the second question. About the tumor microenvironment. The first one is about the BATF3 intrinsic expression on immune cell components. It's better overexpress BATF3 on disease or CD8 T cells to improve cell therapy.
Yes, I mean that's an idea, right? So to potentially play with that molecule. Of course, if you express BATF3 in CD8 T cells for cell therapy adoptive transfer approach, it's better to have a safety switch, because you don't want to have those cells surviving forever. So you need the cells only for the moment that the tumor cells are there, and then later on it's better to switch them off. So yeah, I would say it's something that there is a big potential. So now I think I see the question here as well. Oh, it's better to express on GCs or CD8. Oh, I didn't get the GC. Yeah. I don't know. I think I would go. I mean, CD8 T cells are just easier to play with than myeloid cells. I wouldn't go for DCs, because DCs, BATF3 expression DC seems to be associated with the development, and without I don't know. I mean, I never heard any results that when you overexpress BATF3 in DCs, that the DCs will survive more. But this is associated with the development. As I said, the CD8 T cells, they exist without BATF3. So, but the BATF3 is just regulating the immune response, not the development of the cells. I wouldn't do that in DCs unless we have a very good argument, but I don't see that. And let's say the tertiary, the second question. So the second about tertiary lymphoid organs associated with immune exclude. Dependent mechanism. Well, no, I never heard about that. The only thing I know about the BATF3 in DC1s is that those cells they require BATF3 to develop. What I know is I think if you put some inflammatory condition in place, that might be this thing you're talking about. I think there is a paper that they use, even I think it was Leishmania or Toxo, and they could kind of overcome the absence of the BATF3, and some of these Cs got functional. But I don't know if exactly what you're talking about here. Yeah, I don't know if I answer all the questions. Tertiary lymphoid organ associated and exclude. So the question about the tertiary lymphoid organs, so it's related to the overexpression of BATF3, CD8 or DCs, or to the mechanism independent of BATF3. I didn't get it. Is there another question?
I think we can wait for Vinicius to say in the chat if he have. It's about the mechanism of cross-presenting.
It's cross-presenting. Yeah. So, but what the question was, if it's a BATF3 independent, is it the BATF3 independent in a cross-presenting? Yeah, that might exist, but I'm not so familiar with. Yeah.
Thiago, do you want to ask a question to Marco?
Yes, can you hear me?
Yes.
So Marco, very nice talk, very nice amount of data, very interesting. So the unconventional T cells activation is kind of like a black box for, like a lot of people. So I was wondering if there's any crosstalk between the unconventional T cells and the regular conventional alpha beta T cells for the phenotype you saw in the second part of your talk. And I asked that mostly because you do not see IL-17 production from the unconventional T cells when you knock down IL-23 receptor, right? But it's kind of, was it like a conditional knockdown of IL-23 receptor, or was it, like in all cells?
No, that was a full knockout. Should remember, yeah, that we have the Th17, the conventional CD4 Th17, and those cells they also produce IL-17, but they need to become Th17. So it's gonna take a while. So you need to get activated, clonal expansion. What we're talking about here is few hours, 3 days, sometimes even less. And those cells, they are the first line. So the unconventional T cells, as I said initially, they leave the thymus pre-activated. They have a memory phenotype. If you look at the CD44, which is a kind of marker for activation, they are super high for CD44. And other markers, they express high levels of CD25, L2 receptor that we think that only Tregs express. No, if you check the expression of CD25 in those cells are even higher than in Tregs in the mouse setup. So yeah, that's why when we have the knockdown for the knockout L23 receptor, we still see the phenotype. But this is a part of the innate immune response, I would say. If you wait a couple of days, you might get the Th17 developed. So, and that will also impact the absence of L23, of course, will impact the Th17, the classical CD4, because they also rely on L23 receptor. So that's why, I mean, those experiments, they were done in a short time, because otherwise you start getting the adaptive immunity. I don't know if I answer your question that regard, but that's the relationship. I think they are the first. I would say that those are the first line. As initially I showed, also the group shown for the role of NKT cells in the mediastinal lymph node producing IL-4. We know that later on TFH we also play a role in the germinal center formation, but those cells seems to be there at the beginning and dictating the different immune response in different lymph nodes to start with. And then we could take advantage of those cells in terms of immunization, or in a specific autoimmune disease that you have a high amount of 17 being produced. Those cells are the initial players. So maybe if you switch them off, you have a better outcome. And maybe the conventional T cells that will come later will not drive the whole pathology. I'm just speculating in terms of autoimmunity here.
Yeah, wonderful. Thank you.
Yeah, no problem.
Miriam?
Hi, Marco. Thank you for your talk. It was really amazing. We have a couple of questions here. One by Lucas, who's our PhD student. And first of all, it was really impressive the mouse that you used, that you can do the UV lighting and then see where the cells go. That was really cool. Such a nice tool. So Lucas is going to ask the question. We're doing the mic so we can do wherever we can. You can sit.
Yeah.
Marco, your presentation was very nice. I was very interested in particularly in the second part of it about the unconventional T cells, because after a while you showed that it seems like all three types of unconventional T cells, like the NKTs and gamma delta and mates, they all act kind of the same. And then because I started to wonder about their origin in the thymus and whether they come, like, what would be the evolutionary advantage of having three different somehow populations that act kind of the same, and also it seems like they're almost a bit redundant in some situations. You show that NKT cells are important for IL-4 production, but in some instances it seems like the site of proliferation and the site of stimulus is more important than the phenotype, the cell subtype itself. So because you show that they all behave in a similar fashion when you do one stimulus in one region and then other stimulus in the other. So I was wondering whether what would be the advantage of generating such, in theory, different cells, and then they act kind of the same? Do they actually have very specific differences between gamma delta T cells and NKT cells that it didn't show? I don't know what would be the difference, or should we all group them together in ways? And I don't know.
No, sure, no, yeah, that's a great point. I don't know if I have the answer for this, but first of all, I think that might be, as we defined, based on our single cell sequencing, we could define a very homogeneous niche. We call them this CXCR6 integrin beta 7. But and then you have the whole population of unconventional T cells among those very homogeneous CXCR6 integrin beta 7, just phenotypically speaking. But there are other cells, unconventional T cells, that they don't fall in this homogeneous population. That's another thing. I don't believe that that all of those cells are, compensating each other. Maybe some particular niche that we found, the TH17-like. We also have done this for the TH1-like. So what I attempt to say is that for the innate immunity part, so those cells, they are unconventional. They play a big role in the innate immunity and also in the active immunity, as I've shown a clone expansion for gamma delta, and also in terms of the NKT cells helping to generate some center formation. But the innate immunity part, I think, in general, they are overlapping. And I think the advantage, one possible advantage is that, as I showed you a paper with patients with a mutation in a allele of MO1, though this patient didn't have the innate cells. But the other cells, they're taking over. So they're not suffering, with infection, probably. So the big question here, we tried hard. We generated a triple knockout. We generated the CD1D knockout, the TCR delta knockout, and the MO1 knockout. So we have the triple knockout. Problem is, remember that I showed you in the end, there is a population that we don't know how to classify, because I could stand for gamma delta, I can stand for CD1D, tetramer. I also can stand for innate cells, but there is still 20% of the cells that I cannot stain for any TCR, because we simply don't know. And then what happened with the triple knockout? They have this CXCR6 integrin beta 7 population. Yeah, the population is still there, it's still compensated. So the big question is how can we get rid of this whole population? Then we will understand why those cells are compensating each other. So the best way I found was having the IL-23 receptor knockout. But then I get restricted that idea of fighting bacteria initially, getting neutrophils. That's what IL-17 usually does. But I didn't find a way. I just got rid of the cells functionally, but only that kind of function, IL-17 production. But what would happen if you have a mouse or a mouse line that population does not exist? So this mouse line does not exist. That's the main issue here. So then I don't have the answer for you because we don't have the tools to explore it. But I believe that would give an advantage in terms of innate immune response, mainly. Yeah.
Shouldn't do you think or have you checked whether rag knockout, FOXO1 knockout mice or nude mice lack these cells, and whether you could maybe like use one of those animals and then repopulate them with only T cell progenitors that are already compromised with CD4 mid-high populations, CD8 mid-high populations that wouldn't generate those subpopulations, and then you, in theory, should have a mouse line depleted of gamma delta and mating T cells.
Yeah, the point, those mice, they have a lot of innate lymphoid cells. We thought about it. Yeah, it wasn't easy to perform such an experiment. Yeah, that's an idea, but you also have to get to the amount of innate lymphoid cells just very different from the normal. And then we don't know, and it seems that those innate lymphoid cells somehow, like LC3s and produce L17, they also compensate a bit. Yeah, that's not a bad idea, but it doesn't give us a clear answer. Yeah, we tried that, but the cell, it's difficult to get the cells, proliferating homeostatically. You can't reconstitute the mice in a way that you could, better understand what is the big role of those cells in general. So that's true. We don't know much. What is the role of the unconventional T cells? What we do know is in some specific setup where you have the adaptive immunity, as I showed you, expansion of gamma delta T cells. So that's easy because it's antigen specific. But in that situation that you have a huge overlapping and the cells working as functional unities, it's still not so easy to prove why they are there, why they are compensating each other. That's what we found that is more relevant in that study is that it seems that every lymph node is different, although they have those the same unconventional T cells, but transcriptionally speaking, they are different. And they might get that information from the tissue early on during the development when they are leaving the thymus, going to the peripheral tissue and getting inputs there, and then migrating constantly to the bacteria.
Thank you.
Yeah.
So just continue. There's a couple of questions. I'll try to make them quick, but just continue on that that tone that Lucas was bringing. The first thing I thought was that was a form of coordinating responses, since the antigen that activates each one of those unconventional T cells is different and sometimes even categorically different, right? So NKT cells may be glycoproteins, and gamma delta T cells may be whole proteins, free whole proteins or something like that. So it seemed to me there will be something that would give us signature of response when you have a complex antigen pathway form of activation, which is most what would happen in the wild, right? As we were selected evolutionarily, so we wouldn't be able at the time that we would die or live. I guess we wouldn't have specific only protein antigens or only protein antigens. Would always be something bigger that will be more complex, and that you would need some sort of a coordinated response. So I thought that what you were showing very nicely was that you could coordinate differently each lymph node such that you'd have a targeted response. And that fit really well with some previous data that if you take dendritic cells from the mesenteric lymph nodes or the spleen and would activate T cells in vitro, the T cells would return to the organ where the dendritic cells were originally extracted from. So I thought it was showed in a very beautiful way that you could get specific niches from each one of the lymph nodes such that it would be let's say, specialized in dealing with the type of response that would come from that place that you're draining from. I'm not sure if that's what.
Yeah, no, that's it. And, you know, the biology of DC actually inspired us very much in that regard to better understand, you know, the unconventional T cells. I mean, this project started somehow. You know, we are always we found a new population. We found a new unconventional T cell. I didn't show you here, but it was so hard to define what is the TCR of this cell population. Then we switch gears and say, oh, that might be not the way to work with those cells, to understand those cells. They might, you know, they might play a different role depending on the lymph node. And then we figure out then that they could also behave as a functional unit. But I agree with you. I think that the main message here is it's completely unfinished study. As I said, it's a novel concept and a lot of caveats. But I think what is clear is that every lymph node is different, and then we can potentially take advantage of this information to you know to address problems that we want to address in terms of autoimmunity or vaccine or even cancer therapy. Yeah.
So two very quick points. Have you looked at QA1 restricted CD8 T cells in terms of the second part of your talk? And on the first part of your talk, have you ever found a naturally occurring human tumor where BATF3 was upregulated? Like, because it looked like an oncogene.
Yeah, it is. Yeah. No, I mean, I never checked that. I never checked that. I didn't have the chance to see this. That's why, you know, it's yeah, that was our concern. I'm starting from your second question. So that was our concern. I mean, we're just doing basic research. We speculated BATF3 might be very interesting, increase the T cell stamina capacity. But that was a concern because that molecule could be an oncogene. And but then with this TAC2 work that came out, I mean, that's also associated with genomic stability. But it seems that when BATF3 is constantly expressed in CD8 T cells, it's somehow beneficial. But yeah, we have to have a way to put um, you know, a safety switch in the system if we want to export that. Sorry, your first question was about the MHC, which kind of MHC
The
restriction?
first was the QA1 restricted in the mouse or HLA-E restricted in humans. That is unconventional CD8 cell, but it still like develops very similarly to the conventional CD8 cells.
Mm-hmm.
And they can play a role deleting CD4 T cells. So I'm not completely sure if it fits the topic that you were addressing, but they might in conceptually, right? Since they're not acting as a unusual way. Yeah.
No, I agree with you. No, and we know I never checked that, but I agree there is a potential that they also fall in the same conceptual idea. But I never checked. I don't have data on it. I don't know.
Okay, thank you very much, Marco. I would have like some other questions, but I think we're already like going. But thank you very much, Lisa, as well for the invite. It was amazing to hear your talk. And I think Ana Carolina congratulated you as well in the chat.
Oh, thank you. Yeah, I saw it here. Thank you, guys. It was great to be with you. I'm also have to go. I'm late for a meeting here. So thanks for the invite, everybody. It was great discussion. I hope to see you soon somewhere.
Muito obrigada, Marco.
Nada, gente. Até mais.
Tchau, tchau.
Tchau, pessoal. Até semana que vem.